So now that we understand a little bit about DNA structure, let's talk about how DNA copies itself. It's important to get the biological information out of that molecule, but it's also important to copy that information every time we want to make a new cell. And so that's what we need to take a look at. So we'll look at DNA replication. The concept of DNA replication is not new. It goes back to 1953, right after Watson and Crick came out with the structure of DNA. They proposed their idea for how DNA must copy itself. And their model that they used was called a semi-conservative model. In a semi-conservative model, what they mean is that they said is that DNA opens up and unwinds. The exposed bases then are matched up, and then at the end of that you have two new molecules, half of which belong to the original molecule. So half of each molecule, or half of each new molecule, was conserved from the old molecule, semi-conservative. There was a lot of disagreement about that. There were like three different camps as far as DNA replication goes. Max Delbruck, who was one of the founders of molecular biology, big, huge name, like a celebrity in science, and he said basically, no, that can't possibly be right. That's like trying to keep, that's my thread analogy, trying to keep two pieces of thread as long as a football field from getting tangled. He said it couldn't possibly be that. That's too difficult. He and others believed that DNA replication was conservative. And what conservative means is that the DNA molecule itself is a template, and somehow your cells build an exact copy right next to it, so that at the end you have two copies of the DNA, and one is the original and one is brand new copy. And then still there were other people who thought that DNA replication was dispersive, which means that the DNA molecule is like shattered into pieces. Each piece is copied, and then somehow the molecule is stitched back together. Two copies of it are stitched back together from whatever pieces happen to be lying around. That seems to be an inefficient way of doing it, and in fact it is. So this is a little diagram to show how the different models would look. And so in the top semi-conservative, you'll see the original molecule is in blue, and at the end are two daughter molecules. Each have one blue strand and one red strand, the red strand being new. The conservative model in the middle, our original model is blue. Again, we still have the original model, and then we have a brand new copy of it. And then the dispersive model, the original molecule is blue, the new stuff is red, and you can see how each of the two copies are kind of mishmashed between the two. So we need to figure out which of those it is, because without any experiments, we don't have any basis for claiming one method over the other. And so Delbruck said that we should maybe radioactively label the DNA and see what happens, but it didn't really work. There wasn't any real strong result. So we needed a new technique to figure out how DNA was going to replicate. And Matthew Meselson and his co-workers figured out a way to do it. And it's called the density shift experiment. Now, I'll explain the density shift experiment so that you understand what's going on here. You may need to review a little bit as far as what you remember about isotopes, because the concept of density shift also works on isotopes, but instead of using radioactive isotopes, which give off radiation, Meselson used non-radioactive isotope. He used something called heavy nitrogen. So real quick reminder, nitrogen normally is nitrogen-14. That's the isotope that's the most common, and that means it has 14 total protons and neutrons in its nucleus, 7 of each. Well, there's an isotope of nitrogen called nitrogen-15, which has an extra neutron. And because it has an extra neutron in its nucleus, it's slightly heavier than normal nitrogen. So what Meselson did was he grew up some bacteria in food, medium food, that only had heavy nitrogen. And since nitrogen is such a huge part of DNA, once those bacteria divided and produced new bacteria, and they divided and produced new bacteria, eventually all of the bacteria in that petri dish would have DNA that was heavier than normal DNA because it carried this heavy nitrogen. So they had heavy DNA. When he broke open the the bacteria, and took their DNA out and spun it in a centrifuge, heavy DNA goes to the bottom. Okay? Now, the shift of this experiment, and why it's called the density shift experiment, is because what he could do was he could compare that to bacteria that were grown in normal food source that had normal nitrogen-14, regular or light nitrogen. And when those bacteria were broken open and their DNA was centrifuged, the band was close to the top of the tube. So we have sort of a standard. If DNA has only got heavy nitrogen in it, it's going to be a band at the bottom of the tube, and if DNA has only got light nitrogen in it, it's going to be a band at the top. So the way the shift works is this. You take your bacteria, you grow them up with heavy nitrogen in their food source, so all their DNA is heavy. And then you shift them over. You take the bacteria and you move them. And you move them over to a different food source that only contains light nitrogen. And those bacteria, you let them divide. And when those bacteria take up the light nitrogen and they divide, their offspring can then be broken open. The DNA can be extracted and spun down. And when that happens, what you see is a medium band and a light band. You don't see any heavy DNA anymore. So right away, that immediately destroys the conservative method, because in the conservative method, when DNA is copied, you still have the original molecules there. And if we started with heavy DNA and we copied it in a light food source, we should still have the heavy DNA, but it wasn't there. There was no heavy DNA in the tubes. So right away we can eliminate conservative. So now that means, is it dispersive or semi-conservative? Well, if we look at a picture of this, you'll understand how this works, but the fact that only medium and light are shown, not just medium, means it was semi-conservative. Here's a picture of how it looks. So we know that if we start with a heavy DNA, the red, and we allow it to divide in bacteria that are only eating light nitrogen now, they've already got heavy DNA, but all the DNA that they're going to make from here on out will only have light nitrogen. If it was conservative, we should have then two density bands after the first division. We should have a heavy band and a light band. And then that will continue on every time we allow it to divide. And that was not what was seen. If we follow the dispersive method, remember the dispersive method, we start with a piece of DNA, we break it into pieces, we make new DNA pieces to copy those, and then we put them back together willy-nilly so we have some of each. Well, in that case, we should only have one medium density band, if that's the case, because all of the original heavy DNA is mixed in with light DNA, and all we've got is medium mass DNA. In the semi-conservative method, each time we divide, half of our original molecule goes with a new half of a molecule. And so after two replication cycles, if you see it here, we've got a medium band and a light band. And that was exactly what was seen in the results. So looking at the results of the density shift shows us or proves that semi-conservative is the way to go.